growth and development. So in this lecture, we will see that what are the different parameters of growth, how plant growth is different from that of the animal growth, and also we will see that what different types of growth takes place in plant. So starting with the introduction, firstly, we should know what actually is growth. So how do we define growth? Growth can be defined as an irreversible permanent increase. So this is very important point, that it is irreversible and it is a permanent increase in the size of an organ or its parts or even of an individual cell. So it can be defined as an irreversible permanent increase in the size of an organ or its part or of an individual cell. And it is one of the most fundamental and conspicuous traits of living being. So we know there are certain traits of living being. For example, one of them is growth, other one is reproduction, etc. So growth is one of the most fundamental and conspicuous traits of living being, and it can be defined as an irreversible permanent increase. Why we are saying so? Because you might say that there is a heap of dust and with time it increases in size. So can this be defined as a growth? Can this be defined as a growth? No. Why? Because it is reversible and it is not permanent and not permanent. While if we see, for example, the size of a leaf, suppose this is the initial size of the leaf, it grows to a larger size. So in living organism, this is actually the growth, which is permanent and irreversible. So this is the very important point that we need to remember, that growth is actually an irreversible process. It is a permanent increase in the size of an organ or its parts or even of an individual cell. Now, in order to grow, what we need is energy. So generally growth is accompanied by metabolic processes. These metabolic processes can be catabolic or anabolic. So anabolic means that leads to the synthesis of new things. Like for example, if I give you an example of anabolic process where the synthesis is happening, so we can say that different amino acids lead to the formation of protein. And this protein plays an important role in growth. Right? So growth is usually accompanied by metabolic processes like catabolic process or anabolic process, in which anabolic means the synthesis of higher compounds and catabolic means breakdown of compounds into simpler units. So catabolic means breakdown of compounds, anabolic means the synthesis of the compound. These two help in providing the components and the energy for the growth. And therefore, we can say that growth occurs at the expense of energy. Therefore, for example, the expansion of leaf is a growth where we can see that there is an irreversible permanent increase in the size of the organ. Now we will talk, our talk will majorly focus on the growth in the plants. So similar to animals, plants also show hierarchy. So cell is the structural and basic unit of life in the plants. Different structures, different organ system are made up of cells. And tissue, as we know, is a group of specialized cells that work together for a particular function. Then these different tissues lead to the formation of an organ, which is a distinct structure that is made up of different tissues. Like it may consist of a combination of various tissues. Like, for example, if I talk about the cells in plants, so there are different types of cells like parenchymal cells. The other type is collenchymal cells. It can be sclerenchymal cells. So these different types of cells are present in any organ or organ system of plants, and these different cells combine to form a tissue. Now tissue can further be of various types, like for example, it can be dermal tissue system, that is outer protective layer, or it can be vascular tissue system that consists of xylem and phloem. It consists of ground tissue system, etc. etc. So these tissue systems lead to the formation of organ and organ system. And organ system is a collection of organs that carry out specific function within an organism. And finally, leading to the formation of a complete organism. So how do we define an organism? An organism can be defined as a living thing that carries out all of its life functions. So therefore, similar to animals, plants also show hierarchy in their structure. So there is a structural organization in plants also, starting from the cells that can be parenchymal, collenchymal, sclerenchymal, etc. to the leading to the formation of higher structure, which is the organism, that is a living thing that can carry out its normal routine life functions. So the first step in the plant growth is germination. We all might have witnessed this process of germination during our childhood or even during our lifetime. So we all have witnessed this process of germination. That is the first step in the growth of the plants. So after the fertilization of the seed, when it gets appropriate conditions, it germinates. And the further growth will lead to the formation of a complete large plant. So the first step in the process of plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. Like for example, the favorable temperature, favorable light, favorable rain, favorable water conditions, etc. are available for the seed to germinate. Only then, when the favorable conditions will be existing in the environment, the seed will germinate. And the first organ to emerge from the germinating seed is the radicle, which is the embryonic root. So radicle is the embryonic root. That is the first organ that originates. Right? Next, the shoot tip must break through the soil surface. So next what happens is the shoot tip breaks from the soil surface. In garden beans, this is the germination process of the beans. This is the germination process of the bean, which is a dicot. So in garden beans and many other eudicots, a hook forms in the hypocotyle. You can see here a hook-like structure is formed in the hypocotyle. This hook-like structure is being formed in the hypocotyle, and it pushes the growth, and the growth pushes this hook above the ground. So this hook-like structure is then pushed above the ground due to the growth, resulting in the straightening of this hypocotyle structure. So in response to the light, the hypocotyle straightens, the cotyledons separate, and the delicate epicotyle, which is now exposed, spread its first. So this hypocotyle, which is earlier the hook-like structure, that due to the growth, it pushes out of the ground, resulting in the formation of the structure, which is the epicotyle. So hypocotyle is mainly the root formation, and epicotyle mainly results in the stem formation. So now exposed spread its first true leaves, which are distinct from the cotyledons or the seed leaves. So these are the true leaves that are being formed later, which are different from the. So you might the roots, the embryonic roots are known as radicles. Then we have leaves also. The leaves are the true leaves, which are different from the cotyledons or the seed leaves. So in embryonic stage, the seed leaves are known as the cotyledon, and in the larger plant, when the plant completely germinates, the leaves are known as the true leaves. Similar to hypocotyle, similar to radicle, which are the embryonic root. So these radicle is the embryonic root. We also have embryonic shoot, or the rudimentary shoot of embryonic plant, which is known as plumule. So plumule is known as the embryonic shoot or the stem. Right? In most of the cases, if you forget what is radicle, what is plumule, if you get confused between these things, so radicle, since it is starting with R, it is the embryonic root. So if you will remember one, you will be able to remember the other one is for the shoot. So radicle is the embryonic root and plumule is the embryonic stem or the rudimentary stem. So in the absence of suppose in case after the fertilization the seed formation takes place and the certain favorable conditions are not present. So in the absence of such favorable conditions which are required for the germination of the plant, germination of the seeds do not germinate and they go into a period of suspended growth or rest until or unless the favorable conditions came. So once the favorable condition returns, germination starts. Right? So this is the first step in germination where we can see that from a single root, from the single seed, we can see a whole plant grows. So this is by the process of germination where there is a changes in the organs that takes place in the embryonic organs. Like for example, the germinating seed, the germinating seed has a root which is known as radicle, and it has hypocotyle and epicotyle structures that lead to the formation of the root and shoot. Now, there are certain characteristics of plant growth which are different even from the animal growth. So we know that in animals, if I talk about the growth in animals, after a certain age, we see that the growth stops. So such type of growth is known as determinate growth, where after a certain age the growth stops. But one of the very important feature in plant growth is that they are usually indeterminate. That is, it has unlimited capacity to grow. So plant growth is unique in the sense, this can be asked in the exams also, that whether the plant, one of the very important characteristics of the plant growth is that the plant growth generally is indeterminate. Why I am saying generally? Because there are certain parts that undergoes determinate growth that stops dividing after a particular, that stop growing after a particular age, but usually the plant growth is generally indeterminate. So plant growth is unique because plants retain the capacity for unlimited growth throughout their life. The ability of the plant is due to how growth happens. Usually how growth happens when a cell divides into two, right? So due to the cell division is one of the very important feature of the growth. So why they can be having the indeterminate growth? Because they might be having certain cells that have the capacity to divide. So the ability of plants is due to the presence of meristems which are present at certain locations in their body. Now what is the important feature of this meristem is that the cells of such meristems have the capacity to divide and self-perpetuate. So these cells have the capacity to divide. One cell may divide into two, these cells may further divide. So in that way, they have the capacity to divide and self-perpetuate. The product soon loses the capacity to divide, and such cell comprises the plant body. So the presence of meristems that contains the cells that have the capacity to divide and self-perpetuate leads to the indeterminate growth of the plants. And the products which are formed as a result of this division may soon lose their capacity to divide, resulting in a formation of the plant body. And this form of growth, wherein new cells are always being added to the plant body by the meristem activity, is called the open form of growth. So this form of growth is also known as open form of growth, where the new cells are always being added to the plant body. So suppose the growth has already taken place, like the shoot has grown to a particular size, roots have grown to a certain size, the shoots have grown to a particular size, but the apical region, the apical meristems have the cells that have the capacity to divide, that can divide. So it will lead to the continuous growth, right? So this form of growth is also known as the open form of growth. Now, growth can be of two types. So we see that how this growth is different from the animals, because in case of animals, the growth is determinate. That is, the growth is limited to a particular age, but in the plants, the growth is generally indeterminate. That is, it has the unlimited capacity to grow, capacity for unlimited growth throughout their life. And this is because of the meristems. This is because of the meristems. These meristems contains the root. So they can be apical, root apical meristem and shoot. They are present in the root apex and in the shoot apex. So the cells of such meristems have the capacity to divide and self-perpetuate. This is important, that the meristematic cells. So there is one more process I would like to add here, which is, there is one more term that I would like to explain to you all here, and that is the differentiation. So what is differentiated cells? Differentiated cells are the ones that have lose their capacity to divide and have acquired a particular function to be a part of a tissue. Fine. So these cells, which I say that the product who loses the capacity to divide, and such cells comprise the plant body, these are actually the differentiated cells. However, the meristem cells that have the capacity to divide and self-perpetuate are undifferentiated. Undifferentiated. That is, they have not acquired any particular proper function, and their main function is to divide and grow. So this is a very big difference between the differentiated cells and the undifferentiated cells. So differentiated cells have acquired a particular function in a tissue to be a part of a tissue, while if I talk about the undifferentiated cells have still have their capacity to divide and self-perpetuate, and they do not have any other definite function. They divide and grow. So please do not get confused in between the terms like indeterminate or differentiation. So the growth is indeterminate type, that is, the growth is of a type which has the unlimited capacity to grow throughout the life, while this is because of the presence of the undifferentiated cells, that is, the cells that have the capacity to divide and self-perpetuate in the meristems that are present in the root apex and in the shoot apex, helping in the growth of the plants. And this form of growth is known as the open form of growth. So this is about the characteristics of the plant growth, which is very important from this chapter. So growth can be of two types. The first one is primary growth and the second one is the secondary growth. So primary growth and the secondary growth. Primary growth, as the name suggests, it takes place first, follow primary growth. Now, what actually is primary growth? What is the difference between primary and secondary growth? We will see that. So there are two types of meristems. The first one is the apical meristem and the second one is the lateral meristems. As I mentioned, meristems contains the cells that have the capacity to divide. Right? So one is the apical meristem, the second one is the lateral meristem. Apical means on apex, lateral means laterally. Sorry. So lateral means laterally. So apex means on the top and on the root, at the bottom of the root. And laterally means, like for example, if I talk about any cylinder, for example, considering it as a stem, so this will be the apex and this thing will be lateral, right? So if there is a growth in this, that is, if there is a growth in the width region, it is because of the lateral meristem. And if there is a growth in height, it is because of the apical meristem. So if there is a growth in this direction, it is the apical meristem. And if there is an increase in the girth or the width of the plant, then this is because of the lateral meristem. So height is because of the apical meristem. The lateral growth is because of the lateral meristem, right? So what is primary growth? Primary growth, apical meristems which are located at the tips of the roots. You can see here it is a root apical meristem, and then we have a shoot apical meristem which is present on the tip of the shoot. So these meristems have the cells. So and they are also present in the axillary buds of the shoots, provide additional cells that enable the growth in the length. A process which is known as the primary growth. So the growth in the length is known as the primary growth that allows the roots to extend. This shoot apical meristem will lead to the increase in the root size, and shoot apical meristem will increase in the height. So primary growth allows roots to extend throughout the soil and shoots to increase their exposure to light. So this is the primary growth where the which are present in the shoot apical meristem and the root apical meristem. This allows the growth of the shoot region and the root region in the upward and downward direction, resulting in the primary growth in the shoot region results in the extension of the roots throughout the soil and the shoots to increase their exposure to the light. And secondary growth is the growth in the thickness. So growth in the height is usually the primary growth, and secondary growth is the growth in the thickness, which is the lateral, which is because of the lateral meristem. So this growth in the thickness, that is in this region, you can see here by these arrows. So these arrows shows the lateral growth. This is known as the secondary growth because it is caused by the lateral meristems, which are called the vascular cambium or the cork cambium. So these cylinders of the dividing cells extend along the length of the roots and the stems, and the vascular cambium adds layers of vascular tissue called secondary xylem, which is the wood, and secondary phloem. So you might have seen that when the plant starts growing, the shoot is very delicate, very soft, very green. This is because only the primary growth has happened up to that time. But with age, as the plant age, we see that in the plants that last longer, we see that these plants have a layer of wood also. So this is because of the formation of the wood, which is the secondary xylem, due to the secondary growth. So the vascular cambium adds layer of vascular tissue called secondary xylem, wood, and secondary phloem. And the cork cambium, what is the function of this cork cambium? This cork cambium replaces the epidermis with the thicker, tougher periderm. So you might have heard about the cork, which is obtained from the plants. So this cork is nothing but actually the secondary growth of plant, which is happening because of the cork cambium that replaces the epidermis with the thicker, tougher periderm. So epidermis is now replaced by the periderm in the secondary growth. So this is the overview of the primary growth and secondary growth. Primary growth mainly takes place with the help of the apical meristems that are present in the shoot and the root region. And the main function of these meristems is that they provide the cells, helping the division of the cells which are present in this region results in the extension of the shoot in the upward direction and the extension of the root in the downward direction. And what is the function of secondary growth? In secondary growth, it is the increase in the thickness, that is the lateral growth that takes place, and this is because of the lateral meristem which is present in the shoot and root region and the cork cambium which is present. So the lateral meristem actually helps in the also known as the vascular cambium. So lateral meristems that has the vascular cambium and the cork cambium. So vascular cambium results in the formation of secondary xylem and phloem. Secondary xylem results in the formation of secondary like vascular cambium results in the formation of vascular, as the name suggests, vascular. So vascular, what are the vascular tissues in plants? Vascular tissues in plants are xylem and phloem that supplies the water and helps in the movement of the nutrients throughout the body. So secondary xylem and secondary phloem are formed as a result of vascular cambium, and the cork cambium replaces the epidermis with a thicker, tougher periderm. So you can see in this figure, it will make it more clear that in the apex of the shoot region and in the apex of the root region, the shoot apical meristems, the apical meristems are present that at the primary growth, that is the growth in the length. So apical meristem at primary growth, that is the growth in the length, which are present in the apex of the shoot and root, then also in the axillary buds. So these are the three positions. Usually it is easy to remember root and shoot. People forgot that in the axillary buds also the apical meristems are present. So it is important to remember that it is present in three regions: root apex, shoot apex, and in the axillary buds, resulting in the elongation in the height. Now in the woody plants, so it might be asked that in the non-woody plants, so there are certain plants which are non-woody. So the major growth is the primary growth, right? While in the woody plants, lateral meristems add secondary growth, increasing the diameter of the roots and the stems. So we know that, so in the primary growth, if we will see, there is a formation of the center layer is known as the pith. It has primary xylem, then followed by primary phloem. Primary xylem and primary phloem are present. Cortex region is present, and the outer covering is the epidermis. While we know that in the secondary growth, so the lateral meristems can be vascular cambium or the cork cambium. So in the secondary growth in stems, if we will see, we have the formation of secondary xylem as well. So this secondary xylem is the result of secondary growth. The secondary growth has happened because the vascular cambium has added secondary xylem and secondary phloem to it. So primary xylem was already present there. You can see here that the primary xylem was present. So this is the formation of the, so this is the secondary growth. This is the result of secondary growth. Similarly, this is the result of the secondary growth of the vascular cambium. This is the primary phloem. Also in the cortex region, we know that the epidermis region was earlier present above the cortex. So epidermis region was present, but in the secondary growth, during the secondary growth, the cork cambium adds the secondary dermal tissues. Now, what is the secondary dermal tissues? So this epidermis is replaced by the periderm. Right? So this periderm is the result of the secondary growth, which is due to the cork cambium. So this is the structure that denotes the primary and secondary growth and the changes that happen in the during the secondary growth. Now, can we measure growth? So yes. Why? Because even in humans also, you might have seen the growth, how we usually perceive growth. So we usually perceive growth by the height measurement or by the increase by the increase in the lateral body measurement. So growth can be measured. For example, a child at 4 years of age may be 2 feet, 3 feet, but in the later years, it may change up to 4 to 5 feet or 6 feet, etc. So growth is measurable. And even in the plants also, growth can be measured both at the cellular level and even above that. So at the cellular level of growth, if I'll talk about the cellular level of growth, that is the growth at the level of cells, it can be measured by measuring the increase in the amount of protoplasm. So the increased amount of protoplasm, which is the liquid which is filled inside the cell, present inside the cell, increased amount of protoplasm can be directly considered as a parameter to the growth. But it is difficult to measure the amount of protoplasm which is present in the cell. Therefore, we measure the growth by various parameters. For example, we can see increase in the fresh weight, increase in the dry weight, increase in the surface area or volume, or increase in the number or size of cells. So these are the different parameters of growth because we cannot directly measure the amount of parameters of measuring the growth. Right? So it can be increase in fresh weight, increase in dry weight, increase in the surface area or volume, and increase in the number or size of cells. For example, one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in watermelon may increase in size up to itne times, 3,50,000 times. So in the former, we are counting the number of cells. So the cells are being counted in this. So it is in growth here is represented as the increase in the cell number, while in the later one, we are showing the growth or expressing the growth in the terms of an increase in the cell size. So we can measure the growth by both increase in the number or the size of the cells. So it can be measured by both the methods. While the growth of a pollen tube, if I want to see the growth of the pollen tube and measure it, then it is measured in terms of its length. An increase in the surface area denotes the growth in a dorsiventral leaf. So you might have seen that in a leaf, this increase in the surface area is considered as the growth. And growth is measured by an instrument which is known as oxenometer. This is important, that we can measure the growth with the help of an instrument which is oxenometer. Also, these parameters are important, that how can we measure? So we can measure by increasing the fresh weight. Suppose today you weight a leaf that weighs, suppose it weighs 50 mg. Next day you weigh that, and if the leaf from the same plant on some other day measures 150 mg, you may say that there is a increase in the fresh weight. Also, you can see increase in the dry weight. So you may dry out the components, and then you may check that whether there is a change in the weight also. So increase in the dry weight also reflects the growth. Increase in the surface area or volume, as I mentioned, and increase in the number of size. Now, what are different periods of growth? So period of growth is generally divided into three phases, which is meristematic phase, elongation phase, and maturation phase. So meristematic phase, we know that in this the cells must be in dividing stage. Elongation phase, the cell growth results in the elongation of the zone. So we will see we have a zone of elongation also, then we have a maturation phase. So firstly, we'll see what is meristematic phase. So the cells here are in the constantly dividing stage, and they are present at the root apex and the shoot apex, represents the meristematic phase of the growth. Now, what are the important features of the cells which are present in this meristematic phase or in this meristematic region, that is in the root apex or shoot apex? What are the features that allows the constant division of these cells are that the cells are smaller in size with abundant plasmodermal connections. So plasmodermal connections are the connections between the two cells. These are these connections between the cells, between the connections between the protoplast of the cells. Intercellular, that is the space between two cells, are absent, and if present, they are very small. Therefore, the connection is very rich and easy nutrient uptake or exchange takes place. Cell walls are primary in nature during this time. They are thin, they are cellulosic. Cells are rich in protoplasm and they possess large conspicuous nuclei. So they have a large nuclei with a large amount of protoplasm. So these are the cells in this region are characterized by these features, that they are smaller in size, they are abundant with the plasmodermal connections, intercellular spaces are present, or if present, they are very small, and they are usually, the cell walls are primary in nature, they are very thin, they are cellulosic. And the cells are rich in protoplasm and the large conspicuous nuclei is present. The next is the elongation phase. So the cells which are proximal to the meristematic zone. So in this we know that meristematic zone is present in the root apex and in the shoot apex. We have seen this. Meristematic zone is present in the root apex and in the shoot apex and in the root apex. So above that, in proximity to the root apex and shoot apex, there is a zone proximal to the meristematic zone. So this zone of cell division is present above that, or in proximity to this zone of cell division, there is a zone which is present, which is the zone of elongation. This represents the phase of elongation. Cells in this region are characterized by increased vacuolation. So you may see there that in the previous case, there was a large inconspicuous nuclei. The nuclei size was large. Nuclei size large in meristematic zone. While if I talk about the cells in the elongation phase, they have a larger vacuole. So they have a large vacuole with small nucleus. Vacuole size is large. So they are characterized by increased vacuolation, cell enlargement, and new cell wall deposition. So this is the elongation phase. And then we have a maturation phase. The cells more close to the phase of elongation present the phase of maturation. That is this one, the zone of differentiation. Cells in this zone attain their maximal size in terms of wall thickening and protoplasmal modifications, and the cells are differentiated to acquire a particular function. Or we can say that they are in the matured cells, so they are in the maturation phase. Fine. Now, how do we measure? So there is a technique which is mentioned in the NCERT, that is the parallel line technique. They have shown this diagram. Parallel line technique, which is used to detect the zone of elongation. So suppose if these zones are marked as A, B, C, D, E, F, G. So suppose these zones are marked as A, B, C, D, E, F, G. We can see that in the A, B, C, D, E, F, G, as the time passes, you can see that this zone from A to D, that is this zone from this, and this. This zone has elongated the most, while there is a very less elongation in this zone. You can see here now, when we'll draw the lines from the earlier zone to the newer zones which are formed, you can clearly see that this zone has elongated. So this can be defined as the region of elongation or the elongation zone. So zone of elongation. So this is defined by the parallel line techniques, where when we have initially marked these zones as A, B, C, D, E, F, G, and then after the certain period of growth, we'll mark the parallel lines, we'll draw the parallel lines to the corresponding same zones. We will see that there are certain zones that have undergone the elongation, resulting in the therefore showing the parallel line, therefore depicting the zone of elongation. So this zone of elongation can be detected by this technique, which is known as the parallel line technique. So you might be asked by giving this figure that what is being shown in this figure. So what actually they are trying to show is they are trying to show the parallel line technique for measuring the zone of elongation. Right? Now, very important term is growth rate. So we have measured the growth, but when we measure anything with respect to time, if we measure anything, it is defined as the rate. For example, distance traveled per unit time is the rate. That is also known as the speed. Similarly, growth per unit time is defined as the growth rate. Right? This is defined as the growth rate. So the increased growth per unit time is termed as the growth rate. So increased growth per unit time is termed as the growth rate. The rate of growth can be expressed mathematically and as an organism or a part of organism that can produce more cells in a variety of ways. The growth rate shows an increase that may be arithmetic or geometric. So growth rate can be of two types, arithmetic or geometric. This is important, that growth rate can be of two types. The first one is arithmetic, where the growth takes place in the arithmetic fashion, and the second one is geometric, where the growth takes place in the geometric fashion. What do we mean by arithmetic or geometric fashion is that in the arithmetic fashion, it is the linear growth. Like for example, like with time, initially 2 cells, then 4 cells, then 6 cells, then 8 cells, etc. This is a linear growth. But if instead of this, 2 cells results in the 4 cells, these 4 cells then result in the 8 cells, 8 cells then results into the 16 cells. So just doubling is happening at a particular time. So in that case, it will be a geometric growth. So growth rate may be arithmetic, that is linear, while it can be geometric also, which is non-linear, which is a S-shaped sigmoid curve that we will see in this lecture. So why this linear growth is happening? In arithmetic growth, only one daughter cell among the two further divides, while the other differentiates, that is acquire a function and become mature. So differentiated cells do not undergo division, they stop dividing after acquiring a particular function. So in the arithmetic growth, you can see here that the growth is linear. So why we are saying this it is linear? Because suppose in these 3 cells, out of these 3 cells, if I consider this cell as 1, this cell as 2, so this cell 1 has stopped growing. So it has become mature. While if I talk about cell 2, this cell 2 then further divides into the 2 daughter cells, out of which one will then again become mature, and only the 2 daughter cells. Out of these 2 daughter cells, only one will. So you can see here that this has become matured, and now only this one will divide to give rise to 2 cells. So you can see that only 2 cells are being produced. Now, since out of these 2 cells, only 3 cells will be produced. So 1, 2, 3, and 4. The one cell is getting matured and the other cell is dividing. Out of these divided cells, one cell then again become mature and the remaining divide. So in the arithmetic growth, following mitotic cell division, only one daughter cell continues to divide. How we represent growth? We usually represent growth like this, right? Where the new cells are formed, these new cells further undergo division like this. But in this case, what is happening is, in arithmetic situation, if I want to show it like that, so what will happen is, if there is one cell, if suppose there are 2 cells initially, so out of these it will not be able to divide, only it will divide into the 2 cells. Now out of these 2 cells, when they will divide, one cell will become mature, one cell will become mature. Fine. So one cell will become mature and will stop, resulting in the formation of 2 daughter cells. Out of this, one stops dividing, the other one divides into 2. Out of which the one further stops dividing, the other one dividing into 2. Similarly, this thing happens. So it is taking place linearly. Right? So the simplest expression of arithmetic growth is exemplified by a root elongation at the constant rate. So this is happening at a constant rate. So root and shoot elongation at a constant rate is the arithmetic growth. So it is mathematically expressed as LT is equal to L naught plus RT. Now what are these terms? LT is the length at time t. This is the length. So we know that the equation, this you can see here that it is the equation of a straight line where y is equal to mx plus c. Y is the y-axis unit, x is the unit which are present on the x-axis. C is the intercept. Here in this case it is L naught, and m is the slope. That is the rate constant, rate at which change is happening. Fine. So this is the straight line equation, y is equal to mx plus c. So this straight line equation we can relate here. This y is the LT, that is the height of the plant at any point in time t. RT is equal to mx, where t is the unit on the x-axis. So on the x-axis we have time. You can see here that on the x-axis we have time, and on the y-axis we have the height of the plant, which is LT. This is actually the C. Here it is L naught, the initial height. So length at the time 0, L naught, and R is the growth rate or the elongation per unit time. And this is represented by the slope of this line. That is this theta. The curve is a linear curve. The curve is a linear curve. So this is the arithmetic growth rate where the growth is linear, where after division only one daughter cell divides while the other differentiates. The second pattern of growth is the geometric, and arithmetic growth takes place in the helps in the elongation of the root and the shoot. And geometric growth, in which both the progeny cells following mitotic division retain their ability to divide and continue to do so. So considering it as one cell, it divides into two, and these two daughter cells when further divide into the other. So every cell has the capacity to divide. So one cell resulting in two cells then will result in the four. In the previous case, after two, it will be three, then it will be four, suppose. But in this case, what will happen is the four cells will result in the 8 cells instead of 6. And these 8 will further then lead to the 16 cells. So this is a geometric pattern which is being followed. So this is the geometric growth in which every cell, every progeny cells divides and they retain the ability to divide and continue to do so. So early embryonic development, division in zygote is the division in the unicellular organism is the geometric growth. So the early embryonic division is the geometric growth. And it is represented, so how we were representing the arithmetic growth. Arithmetic growth were represented by the equation, straight line equation. But in this, the equation is different. Here W1 is equal to W naught ERT. So W1 is the final size, weight, height, number, etc. W naught is the initial size at the beginning of the period. R is the growth rate, and base of the E is the base of the natural log. So this is an equation of the geometric growth, right? Where W1, here R is the rate at which the change is happening, T is the time. W1 is the final size or the final result, and the W naught is the initial size at the beginning of the period, is the base of the natural log. So in most systems, the initial growth is slow, which is represented by the lag phase. You might have studied it in bacteria also. In bacteria, the growth pattern is the similar type, which is divided into lag phase, exponential phase, and the stationary phase. So most of the growth happens during the exponential phase. In the initial phase, due to the lack of nutrients, less nutrient supply, the growth is slow. But once the number of cells exceeds to a limit that it can now grow indefinitely, so the growth booms, resulting in the exponential phase, and ultimately due to the when all the nutrients are being consumed by the growing cells, it results in the stationary phase where the growth terminates. So it increases rapidly thereafter at an exponential rate, which is the log or the exponential phase, and it is also known as the grand phase of the growth, because majorly the growth happens during this phase. However, with limited nutrient supply, the growth slows down, leading to a stationary phase or the steady phase. So initially, since the number of cells are very few, like there is only one cell or two cell considering the initial number of cells, therefore the growth is really low because of the less, I want to make some correction here, because of the less number of cells initially. Less number of cells initially, the growth is slow. This is very important. Why we are experiencing this lag phase? Because there are less number of cells. But once they reach to a certain number of cells, as I mentioned, the growth will boom. But the stationary phase happens because of the less nutrients, due to the less availability. Now, if we plot the perimeter of growth against the time, we will get a typical sigmoid or S-shaped curve. You can see here it is a S-shaped curve. This curve is known as the S-shaped or sigmoidal curve, and it is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissues, and organs of the plant. Here R is the relative growth rate and is also the measure of the ability of the plant to produce new plant material, and therefore it is referred to as efficiency index. So R is nothing but the growth rate, relative growth rate, and it is also referred to as efficiency index. This is important. Just by giving the formula, they may ask whether what is the correct formula and what is the correct notation. So in this, we see that W1 is the initial phase and R can also be called as the efficiency index, because it is the one that is defining the efficiency based on the R, which is the growth rate. The efficiency of the growth is being defined. Now, the growth, as I mentioned, can be arithmetic or geometric, and the comparison of growth can be done. So, quantitative comparison of the growth can be done in the mathematical form, and it can also be measured, can be done in two ways. Like it can be absolute growth or there can be a relative growth. For example, if I want to explain you about the absolute or relative growth, I may give you an example. Like suppose there is a person A and there is a person B. So, if person A weighs 40 kgs, if person B weighs, if person B weighs 50 kg, and after a certain period of time it weighs 45 kg and it also weighs 55 kg, then obviously there is an increase in their individual weights. And when I am talking about individual, I am talking about the absolute growth. Total growth per unit time, if I am taking the total growth per unit time. So the total growth per unit time in this case is also 5 and in this case is also 5. Therefore, the absolute growth is same in both of these organisms, but the relative growth. So how do we see the relative growth? So suppose in 1 year, the weight increases from 40 to 45 and 50 to 55. But if I talk about, so I'll explain the relative growth with the help of this example. It will be easy for you to remember. So as I mentioned that you get the concept of absolute growth, right? In which both the changes happen in both by the same unit. So in this, the absolute growth is the total growth which is measured per unit time. So the total growth is of the total gain is of the 5 kg, irrespective of what was the initial weight. We are not considering the initial weight, we are just considering the differences of the initial and the final. So absolute growth just means the difference between the final minus initial, right? So how much absolute growth has happened in this? From 50 centimeter square, the area has increased to 55 centimeter square. So it is 5 centimeter square. And in this also the 5 centimeter square area has, like, the increase in the area has happened. So both of them are having the same absolute growth. Same value of final and initial, like the difference is same in both, therefore they have the same absolute growth. But the relative growth is different because the relative growth depends upon the initial parameter as well, that how much the changes happen. You can see here, if I shade this, we can clearly see that the shaded area of this is more than the shaded area of this leaf. That is, although it has increased 5 centimeter per square in the same time, but the relative growth of this larger leaf is slow and the relative growth of this smaller leaf is fast. Why we are saying so? Because relative growth can be measured as the change divided by the initial. So relative growth can be measured as the growth of the given system per unit time by the initial parameter into 100. So the changes of the 5 centimeter per square, the initial parameter is also 5. So it has changed up to 100%. Like there is a 100% growth. The size has just become double. While in this case, if I talk about the relative growth, the change is 5 centimeter, but the initial is 50 centimeter square. So it is just the 10%. So the second leaf has just grown 10%, while the first leaf has grown 100% of its initial size. Therefore, the relative growth, if I measure, it is having a faster growth as compared to this one. So this is how we can quantitatively compare the growth. And now what are the conditions for the growth? As I mentioned, there are certain nutrients, water, oxygen, nutrients. In addition to this, optimum temperature is required. Any deviation from these conditions will hinder the growth and could be detrimental to its survival. Environmental signals such as light and gravity also plays certain roles and affect certain phases or stages of growth that we will see later in the next lecture, that how light and gravity is also affected. So phototropism you might have seen because of the light. So plants are sensitive to such environmental conditions also. Water, oxygen, and nutrients are very essential for the growth. Plant cells grow in size by cell enlargement, which in turn requires water. So this cell intake water and grows in size. Due to the growth in size, cells become turgid, that is tough, and it helps in the extension growth. This plant growth and further development is intimately linked to the water status of the plant, and we know that this only results in the beginning of germination. Also, the water is required for various enzymatic activities which are needed for the growth. So oxygen helps in releasing the metabolic energy and it is required for various growth activities. Nutrients like various macro and micronutrients are required by the plants for the synthesis of protoplasm and they act as a source of energy. In addition, every plant organism has an optimum temperature on which it can grow best, and any deviation may lead to the detrimental situation for its survival. So this was all about the growth, and in the next lecture we will see about the development, that how the development happens in the different parts of the plant. So thank you for attending it.